There are a myriad of different B cells in the human body. Each of the B cells expresses antibodies specific to one antigen. B cells that recognize an antigen proliferate and secrete antibodies. They differentiate to plasma cells, which secrete a large number of antibodies. The secreted antibodies circulate through the body. When they encounter the specific antigens, they bind to the antigens and induce an immune response. The antibody protein consists of heavy chains and light chains. The genes encoding antibody, or known as immunoglobulin genes, consist of one pair of heavy chain gene, called IgH, and two pairs of light chain gene, called IgK and IgL. The heavy chain gene consists of dozens of V, D, J fragments and C fragments. Each of the V, D, and J fragments is rearranged one by one to express the heavy chains of an antibody. Like the rearrangement of heavy chain genes, each of the V and J fragments of light chain genes are also rearranged for expressing the light chain proteins. The antibody, combined form of heavy and light chain protein, recognize one antigen specifically, depending on which fragments were used to rearrangement. If the first recombined and expressed heavy chain protein is abnormal, B cells use the heavy chain gene of another allele to make a new heavy chain protein. Similarly, for the light chain counterpart, one of kappa genes is rearranged first to express a light chain protein. If the light chain proteins expressed in two kappa genes are abnormal, the lambda gene is rearranged to make a light chain protein, completing an intact antibody. Based on the characteristics of allelic exclusion related to the development of antibodies in vivo, HuMab is developing Synthese platform that enables production of more diverse antibody drugs. While human antibody genes exist as homoallelic in conventional transgenic mice, they exist as heteroallelic in HuMab's platform because each allele is derived from the different human races. Synthese can produce superior antibody drugs effective against a wider variety of different antigens.